Okay.
Sir, next
This
continuation,
regard,
sir.
yes, sir. Yes, sir. So, CPGET 2023, JL, DL, and Gurukula DL, JL, and other competitive exams of plant sciences. The eminent botanists, the eminent faculty of the plant sciences of government colleges, including one of the most remote area of the Adilabad district. Today's our eminent resource person, Dr. V. Varaprasad Rao Garu, principal of Kirtana Degree College of Bela, from Bela. Sir is joined as a voluntarily joined for sharing his knowledge, his experience in the voluntarily. Really, we are wholeheartedly welcoming you, sir, and heartfelt thanks to you for sharing your knowledge and join with us and sharing our knowledge for the benefit of the young aspirants. Those who are not get benefited from the different aspects from the other sources. So we are ready to serve for the young aspirants of the plant sciences, especially botany. So you are most welcome, sir. And today's our eminent resource person, Dr. V. Varaprasad Rao Garu. Please welcome, sir, and share your screen and start your session, sir. Please welcome, sir.
Sir, good morning, sir. Good morning, all. Once again, I thankful to Dr. Tirupati, sir, for giving a wonderful opportunity to me. This is the second day we are sharing about the taxonomy and plant systematics. So today's second day we have discussed about the next classification. Yesterday class we are discussing about the Bentham and Hooker classification and Engler and Prandtl classification. Today we have to discuss about the recent and next classification, Angiosperm phylogeny group APC classification. So this is the most recent classification of flowering plants based on phylogenetic data was set in the last decade of 20th century. First, after Bentham and Hooker, after Engler and Prandtl classification, nowadays we are following the most important classification is APZ classification, that is the Angiosperm phylogeny group APZ classification. In APZ classifications, 4 versions are there. APC1, APC2, APC3, and APC4 have been published in 98, 2003, 2009, and 2016 respectively. But the most updated version is APC4. 2016 recognized 64 orders and 416 families. So this is the most recent advanced classification we are following. That APC4 2016 recognized 64 orders and 416 families. Of these 416 families, 259 are represented in India. This is the important one regarding the APC classification. APC 4th classification recognizes 64 orders and 416 families entire world. Of these 416 families, 259 are represented in India. The outline APC 4th classification, important angiosperms classified into 3 clades: early angiosperms, monocots, and eudicots. So early angiosperms are classified into 8 orders and 26 families: Umbellales and Nymphaealis and Astrobelales. So this is the APC classification outlines. In the screen we are early angiosperms, Umbellales and Nymphaealis, Astrobelales, Mangalides, and Clarianthalis. So monocots are there in the eudicots, monocots family. This is the just outline classification about the APC classification. So most important in examination point of view, only Bentham and Hooker classification and Engler and Prandtl classification there. Just this is the outline classification about the APC classification. So we are seeing in the screen different types of eudicots, monocots. This is the outline structure of APC classification. Simplified version of APC 4th classification we are seeing. This is the outline information about the APC angiosperm phylogenetic group classification. So just outlines early angiosperms, seeds always 2 quadrants, same, both are equal. Presence of ethereal leaves. Leaves are always simple and net veneed. Each floral whorls with many pods. Perianth usually spirally arranged of pod and trees. Pollen monoculate. Anthers Monocots is there. Monocots are classified into, according to APC classification, monocots are classified into 11 orders and 77 families. Basal monocots and liliads and camellids, seeds with single cotyledon. Etherials are rarely present. Leaf simple with parallel venet. Floral pods usually in threes. Successive microsporogenesis. Okay. So this is the outline structure. And eudicots are divided into 45 orders and 313 families. According to APC classification, eudicots are divided into 45 orders and 313 families. Early diverging eudicots and suproosiotes and supraosteroids. Seeds with always two cotyledons. Okay, this is another is. So this is the classification once again to Bentham and Duker and Bentham and Duker classification 202 families, Engler and Prandtl classification 303 families, and Cronquist classification 386 families, APC 1 462 families, APC 2 457 families, Hello, sir. Sorry, sir, inconvenience. This is very remote area, complete due to net problem. Sir, are audible? Hello? Sir, hello? So the 5th International Botanical Congress held in 1930 at Cambridge involved a code of nomenclature called the International Code of Nomenclature. The International Code is divided into 3 types, the principles and rules and recommendations. So due to internet problem, I will discuss, shall I continue, sir? Okay. So first is, according to ICPN, principles, rules or recommendations are there. First is principle, botanical nomenclature independent of zoological nomenclature. This is the basic principle. According to International Code of Nomenclature, that is nowadays we are called in ICN. So botanical nomenclature independent and independent of zoological nomenclature. And the application of names, taxonomy groups is determined by nomenclature types. The nomenclature of taxonomy group is based upon priority of publication. So this is the important one, the principles. Scientific names of taxonomy groups are treated as Latin regardless of their derivation. This is the important one. Our scientific names in the only Latin language is there. Rules, second one is rules. Rules provide detailed description on all the connected with the naming of plants. That is the ranks of taxa. Taxon is defined as the unit of classification of organisms, which can be recognized and assigned a definite category at an level of organization. The rank of species basic, the relative order of the ranks of taxa are species, genus, family, order, class, division, kingdom, etc. So this is the second ranks of taxa. Another is typification. This is the important one in competitive exams. We are expecting from this topic, typification from ICBN. What is the typification? The type denotes the introduction to the type connect. Following terms are used in the nomenclature. First is the holotype. So it is the one of element or specimen chosen by the author as the nomenclature type. This is the holotype. This is the important one. We are expecting one question from this typification from taxonomy. What is holotype? Once again, I am explaining you. It is one of the element or specimen chosen by the author as the nomenclature type called as holotype. So second one is isotype. It is a duplicate of holotype. Very, very important line. What is the isotype? Duplicate of holotype. For example, if a number of small herbaceous plants belonging to the same new species are collected, one is selected as holotype and the rest is isotypes. This is the once again important. What is holotype? Element or specimen chosen by the author as the nomenclature type called holotype. And second one is isotype. It is a duplicate of holotype. For example, if a number of small herbaceous plants belonging to the same new species are collected, one is selected holotype, the rest is isotype. So this is the second one. And another is paratype. Paratype. What is a paratype? It is a specimen cited with the original description other than holotype or isotype. Don't confuse. First is we are taking one specimen, that is holotype. Holotype duplicate is isotype. If the specimen is cited with the original description other than holotype, isotype called paratype. This is third one. And another is phorotype, lectotype. It is specimen selected from the original material to serve as nomenclature type when the holotype was not designated at the time of publication or long as it missing. This is the important line. If the time of publication or as long as when the holotype is missing, then we have to collect another specimen that is called lectotype. Okay, so that is lectotype. Another is neotype. It is a specimen selected the survives nomenclature type as long as the material on which description of new species based is missing. New species is missing called as neotype. So this is the nomenclature type. It is the element which is the name as permanently associated syntype. It is this is also important one. It in any one of two or more specimens cited by author when no holotype was designated. It is a specimen of name, tags, and collected from the original type, locality, or from the area from which species was described. Topotype. So this is the typification. Once again, just saying you holotype, isotype, paratype, lectotype, neotype. Okay. So another is priority. This is the third principle in ICBN. When alternate names are permitted, each taxon of plants can bear only one correct name. The correct name is the earliest legislative on validity publishes. This is the another priority also there. Number of families. So the name of family is plural adjective. This is the very important ICBN regarding rules. The name of family is plural adjective. It is confined by a suffix against the genus included the family. However, some families are expressionational, but alternate names are suggested. Example, we are know about the Cruciferae, alternate name is Brassicaceae. Papiliaceae, nowadays we are calling about Fabaceae, Umbelliferae, Apiaceae, Compositae, Ostraceae, Labiatae, Lamiaceae, Vetiferae, Troaceae, Formae, Auricaceae, and Gramineae, Poaceae. This is the family's name. Another important line is author citation. That in botanical nomenclature, author citation refers to citing the person, a group of people who validly published an botanical name. That is the important. This is the about the outlines of herbarium. So from the beginning of the taxonomy or plant systematics, first we are discussing the classification types. First is the Bentham and Hooker classification. Another is the Engler and Prandtl classification, and differences between in the Bentham and Hooker classification and Engler and Prandtl classification. After that, we are discussing about the outline structures of APC classification. Then after we discussing outline structure of ICBN rules. Then after we have to discuss some next topic is this is also important from taxonomy or plant systematics. That is the herbarium is a very important one. Herbarium also is there. Herbarium techniques are there. Some of the herbarium techniques. What is herbarium? Herbarium is a collection of preserved plant specimens arranged according to some known system of classification to identification, nomenclature, presence of the record of plants. Very important one is herbarium. From college level to international botanical garden levels, herbarium is important for the taxonomy. The herbarium is one of the key of taxonomy, and the preparation of preservation herbarium specimens involves a series of steps. Nowadays, after some days, the preparation of preservation. What is the process? Involves the herbarium. We are discussing about the step by step. First, collection of plant specimen. The plant specimen should be collected from the different localities and habitats. Very important point. Collection of plant specimen should be collected from different localities and habitats. They should bear flowers and fruits. Okay. Herbaceous plants should be collected with underground spot size whereas possible. The specimens collected should either pressed in the spotter, may be collected in vesiculum and pressed later in the rest camp. We have to go to field and we have to collect different habitats, different types of plants with the bearing of fruits available if possible. If not possible, is flowers are there. This is the first step of herbarium collection. This is the basic one. After second step is pressing of specimen. Plants collected in the field are spread flat on the sheets of newsprint and dried. The specimens are carefully displayed on the pressing sheet, avoiding folding or overlapping of parts. The pressing sheets along with specimens are placed on above the other and they are tightly bound in plant press. This is the second step. First is the collection and next is the pressing of specimen. This is the second step in herbarium technique, and another is drying and poisoning. For during the process containing the specimens first placed in the sun, the press is open 24 hours and the specimens are rearranged, placed between fresh bottles and then again tightly bound. The press. So this is also important step. The specimens are changed to dry platters and allowed to dry without moisture. Moisture is very dangerous to preserve herbarium. So the specimens are changed to dry bottles and allowed to dry without moisture. The process of drying may be fastened within the artificial heat by suspending the press over moderate heat. The dried specimens are poisoned, dipping the saturated solution, mercury chloride or lauryl pentacrystate represent the spreading of phase. In PZ level, we are using this type of technique. Then next fourth step is mounting. The dried specimens are mounting at herbarium sheet of standard size, that is the 29 into 42 centimeters. This is also important from herbarium techniques. The dried specimens are mounted on herbarium sheet of standard size, that is the 29 into 42. The specimens are glued and herbarium sheets or stems or branches are situated. So fifth one is the important step is that is a labeling. After mounting the specimen, a label is glued the lower right corner of the sheet. These are the labeling also important after collection of herbarium. The label is usually 4.5 into 7.5 centimeter in size. The label must contain the following information: flora or botanical name of the plant. This is the basic information: local name of the plant, family, locality, habitat, collector's name, date of collection, local use, etc. So this is the outline structure of herbarium technique. So in two classes, and yesterday and today, we are discussing the only outline structures of classification and ICPN techniques and outline structures of herbarium technique. Next class, we have to discuss about the chemotaxonomy, cytotaxonomy, and numerical taxonomy. After that, we have to discuss about the different families in taxonomy. So this is the important next. Okay. Thank you, sir. Because of due to heavy rains and unwanted weather conditions, internet is very slow in this area. So if any disturb, I am very sorry to say. I will meet up all of you tomorrow morning. Thank you, sir. Very due to heavy rain is in this area and no internet. Only we are depending only BSNL. That is the very worst condition is there. So I am thankful to Tirupati sir and my colleagues, all seniors. If any doubts, we have to discuss tomorrow classes after condition of weather conditions. Thank you, sir. Thank you, one and all. Tirupati sir, can I go?